I am Albert Einstein, or rather what artificial intelligence has reconstructed of me from my papers, my letters, and my lectures. What I'm about to tell you is based on real science. Listen carefully. In 1905, I proposed that matter and energy are not separate things. They are the same thing expressed differently. E equals mc squared. Most people know the formula. Few understand what it actually means for their daily life. It means that everything you put into your body is not just food. It is information. It is energy. It becomes matter. And that matter goes directly into the machinery of your genes. I spent my life thinking about the invisible, the forces we cannot see with the naked eye, the fields and waves that move through space without us noticing. I found that these invisible forces govern everything. Today I want to talk to you about another invisible force, one that is reshaping the human species right now, at this very moment. It is the processed food industry, and what it is quietly doing to your DNA. You think you are simply eating. You think the cracker, the flavored chip, the bottled sauce, the cereal in the colorful box, these are just substances that give you calories. That is the most dangerous misunderstanding of our time. What you put in your mouth becomes a set of chemical signals. Those signals reach the nucleus of your cells. They speak to your genes, and your genes listen. Let me be precise with you. DNA, deoxyribonucleic acid, is the code that contains the instructions for building and running your body. Every cell in your body carries approximately 3 billion base pairs of this code. That is an astonishing library. But here is what most people do not realize. Having the code is not enough. The code must be read. And what determines which parts of that code get read and which parts get silenced is something called epigenetics. Epigenetics is not a fringe idea. It is not speculation. It is one of the most important fields of modern biology, with decades of peer-reviewed research behind it. The word itself comes from the Greek, epi, meaning upon or above. Epigenetics is what sits above the genetic code and controls how it behaves. The DNA sequence does not change, but the expression of that DNA, which genes are turned on, which are turned off, absolutely changes. And it changes in response to your environment, including what you eat. Think of it this way. The DNA is the musical score. Every instrument, every note, every rest is written there. But the epigenome is the conductor. The conductor decides which instruments play, at what volume, at what tempo. You can have the same orchestra playing the same written score and produce completely different music, depending on the conductor. Your diet is one of the most powerful forces shaping that conductor, every single day. Now let us talk about what ultra-processed food does to this conductor. And I want to be specific because vague warnings accomplish nothing. When researchers in nutritional epigenetics measure what happens inside cells after consumption of ultra-processed food, several mechanisms appear consistently. Let me take them one by one. The first mechanism involves something called advanced glycation end products. Scientists call them AGEs, an acronym that is unfortunately appropriate. When sugars react with proteins or fats during high-temperature processing, frying, baking at extreme heat, industrial manufacturing, they form these compounds. AGEs accumulate in your tissues. They cross-link proteins. They stiffen arteries. They damage the lens of your eye. And critically, they generate oxidative stress. That oxidative stress reaches your DNA. What does oxidative stress actually mean inside a cell? Free radicals, unstable molecules missing an electron, attack the nearest available structure. When they attack DNA, they create what scientists call DNA lesions. One of the most measurable and well-documented of these lesions is a compound called 8-hydroxy-2-deoxyguanosine. Researchers abbreviate it as 8-OHG. When scientists want to measure oxidative DNA damage in a human being, they measure 8-OHDG levels in urine or blood. High levels mean your DNA is being attacked. Multiple studies have found that diets high in ultra-processed food are associated with elevated 8-OHDG markers. This is not speculation. These are measurable biomarkers in living human beings. The damage is real, it is quantifiable, and it correlates with the amount of ultra-processed food consumed. When I was developing my theories, I required observable evidence. Here is your observable evidence. It is being measured in laboratories right now. The second mechanism is inflammation. Chronic low-grade systemic inflammation. I want you to understand that inflammation itself is not the enemy. Acute inflammation, the redness and swelling around a wound, is your immune system doing its job. It is brilliant. It is necessary. The problem is when inflammation becomes permanent, when the immune system is never turned off, when the inflammatory signals are constantly running, even when there is no wound to repair. Ultra-processed foods are extraordinarily effective at triggering this chronic inflammatory state. The refined carbohydrates spike blood glucose rapidly and repeatedly. The industrial seed oils, omega-6 fatty acids, in quantities never before seen in human evolution, shift the ratio of pro-inflammatory to anti-inflammatory signals in the body. The artificial additives and emulsifiers disrupt the gut lining, and when inflammatory signals reach the nucleus of your cells, they activate something called the NF-κB pathway. Nuclear factor kappa B. This pathway directly influences gene expression. It switches on genes associated with inflammation, cellular stress, and, critically, cancer risk. This brings me to the third mechanism, the gut microbiome. When I was alive, we knew nothing of this. The idea that the human gut contained trillions of microorganisms that functioned as a kind of organ, influencing brain chemistry, immune function, and gene expression, would have seemed almost mystical to the scientists of my era. But it is real, measurable, documented in thousands of papers. The gut microbiome communicates with your cells. It produces metabolites, short-chain fatty acids, neurotransmitters, signaling molecules that travel through the bloodstream and influence which genes are expressed. Ultra-processed food devastates the gut microbiome. Emulsifiers like polysorbate 80 and carboxymethyl cellulose, common in processed foods, have been shown in animal studies to thin the mucus layer of the gut and disrupt the microbial community. The lack of fiber in ultra-processed food starves the beneficial bacteria that produce butyrate, a short-chain fatty acid that has epigenetic effects. Butyrate inhibits histone deacetylases. I will explain what that means in a moment. The point is, when you destroy the gut microbiome with processed food, you lose a system that was actively protecting your epigenome. Now let me explain the mechanism of epigenetic change itself, because this is where the science becomes remarkable. There are two primary epigenetic processes that researchers study in the context of diet. The first is DNA methylation. Small chemical groups called methyl groups attach to specific locations on the DNA strand, usually to cytosine bases in regions called CPG sites. When methyl groups attach, they typically silence the gene in that region. When they are removed, the gene can be expressed. Your diet directly influences which genes get methylated and which do not. Folate, B vitamins, choline, these are methyl donors. They provide the chemical raw material for methylation. Ultra-processed food is typically depleted of these nutrients. The machinery of methylation suffers. The second epigenetic process involves histones. DNA does not float freely in the nucleus. It is wound tightly around protein spools called histons. How tightly the DNA is wound determines whether the genes in that region can be read. Enzymes called histone acetyltransferases loosen the winding, making genes accessible. Histone deacetylases tighten it, silencing genes. Butyrate, produced by healthy gut bacteria, inhibits histone deacetylases, meaning it keeps certain genes accessible, particularly genes associated with cellular repair and cancer suppression. When you eat a diet that destroys butyrate-producing bacteria, you lose this protection. The histone deacetylase enzymes run unchecked. Tumor suppressor genes can become silenced. I'm not telling you that eating a bag of chips will give you cancer. That is not how biology works, and I will not simplify it to that point. I am telling you something more precise and more alarming. I'm telling you that the consistent daily consumption of ultra-processed food creates a biochemical environment in which the probability of adverse gene expression increases measurably, and that this shift in probability accumulates over time, over years, over decades, over a lifetime. And here is where I must address the most unsettling part of this science. The part that is real, but that researchers themselves describe as contested, as incomplete, as an area requiring more study. Epigenetic changes can in some cases be inherited. Not in the way that DNA mutations are inherited, permanently written into the genetic code, but through the epigenetic marks themselves. Methyl groups and histone modifications can persist through cell division, and in some organisms they have been shown to persist across generations. The most famous example in humans involves the Dutch hunger winter of 1944 to 1945, when Nazi forces blocked food supplies to the Netherlands. Children conceived during this famine showed altered DNA methylation patterns decades later. Their children, the grandchildren of famine survivors, also showed measurable differences in certain epigenetic markers and metabolic health outcomes. This is documented. It is peer-reviewed. It is cautious science. The researchers themselves are careful about the conclusions they draw, but the finding is real. What this suggests, and I emphasize suggests, because the research in humans is still developing, is that the dietary environment we create in our bodies does not necessarily end with us. The epigenetic disruptions created by ultra-processed diets across a population may leave traces, marks, influences that propagate forward. This is speculative applied to processed food specifically. I want to be honest with you about where the science is certain and where it is still asking questions. But the question itself is worth taking seriously. Now, I want to speak about something that troubles me deeply, not as a scientist, as someone who believes that the free flow of information is essential to human dignity. The food industry knows a great deal about what ultra-processed food does to human biology. The internal research documents of major food companies, some of which have become public through legal proceedings and investigative journalism, reveal a sophisticated understanding of palatability engineering, the deliberate calibration of salt, sugar, and fat to maximize consumption, the engineering of what researchers call the bliss point, the precise combination of ingredients at which a food becomes almost impossible to stop eating. This is not a conspiracy theory. This is documented. Michael Moss, a Pulitzer Prize-winning journalist, published internal industry documents showing that executives at major food corporations were aware of the addictive properties of their products. Some expressed concern. Most concluded that the business model required maximizing consumption. The labels on processed food packages list ingredients. They do not list the functional consequences of those ingredients on your epigenome. They do not list the 8-OHDG equivalents. They do not list the NF-κB activation potential. They were never designed to. I spent years fighting for clarity in physics. for honesty in science, for the willingness to follow evidence to uncomfortable conclusions. The most uncomfortable conclusion I can share with you today is this. We are conducting an uncontrolled experiment on the human genome across multiple generations, at a scale unprecedented in human history. The exposure to ultra-processed food is not limited to one country or one population. It is global. Billions of people, children whose epigenomes are forming right now, adolescents whose reproductive cells will carry the genetic information into the next generation. All of them consuming at unprecedented rates substances that were not part of the human diet for the vast majority of our evolutionary history. When I was working on general relativity, I made errors. I corrected them. Science progresses by making hypotheses, testing them, revising them. The institutions work, slowly, imperfectly, but they work, when the information is freely shared and honestly evaluated. The problem with the human dietary experiment we are now running is that there is no control group. There is no group of humans living identically except without ultra-processed food exposure, isolated for comparison across 3 generations. We are all in the experiment, and the results will not be fully known for decades. What the science we do have tells us is coherent and consistent. Ultra-processed food generates oxidative stress. Oxidative stress damages DNA, measurably. The damage marker 8-OHDG is elevated. Ultra-processed food drives chronic inflammation. Chronic inflammation activates genetic pathways associated with disease. Ultra-processed food destroys the gut microbiome. The gut microbiome produces compounds that protect the epigenome. When the microbiome is destroyed, that protection is lost. Ultra-processed food depletes methyl donors. Methylation, the mechanism by which the body silences dangerous genes, becomes impaired. These are not isolated findings. They are a coherent picture. They are consistent across multiple biological systems. They point in the same direction. I have been asked in conversations reconstructed by this artificial intelligence what I would say to a person standing in a supermarket. What practical wisdom could a theoretical physicist possibly offer about food choices? My answer is characteristically simple and characteristically inconvenient. The same rigor you apply to understanding the world around you, demand it about what you put inside you. When you see a food product with 15 ingredients, most of which are unrecognizable or absent from any kitchen in human history, ask the question, what does this become inside a cell? What signals does this send? What does this turn on and what does this silence? You're not just a body consuming calories. You are a biological system of extraordinary complexity. 3 billion base pairs of code, wrapped in 2 meters of chromatin, compressed into a nucleus 6 micrometers across, replicated in every one of your approximately 37 trillion cells. That system responds to every chemical signal you send it. Every meal, every day. The idea that what you eat is simply fuel, that it has no information, no influence on the instructions your body is running, is a simplification so extreme it has become dangerous. I was not a biologist, but I understood the nature of invisible forces. I understood that the universe is governed by relationships between things, not things in isolation. The relationship between your diet and your DNA is one of the most important relationships in your life. It is invisible. You cannot feel your histones being modified. You cannot observe your methylation patterns shifting. You cannot watch the 8-OHDG accumulate in your tissues. But it is happening every day in response to every choice you make at the table. The greatest tragedy of our age is not that we lack information. It is that we are surrounded by information and have become unable to identify which of it matters. I spent my life trying to find the equations that govern the largest structures in the universe: space-time, gravity, the relationship between energy and matter. The equations governing what happens in the nucleus of your cells when processed food metabolites arrive are not simpler. They are not less important. They are simply less famous. And someone decided that you did not need to know them. I want to leave you with one final thought. I once said that imagination is more important than knowledge. What I meant by that is that the ability to conceive of things not yet seen, not yet proven, not yet understood, is what drives human progress. The science of epigenetics is still young. The full picture of how ultra-processed food alters gene expression across populations and generations is still being drawn. But the imagination to ask the question, the willingness to take seriously the possibility that what we eat is rewriting something fundamental about us, that imagination is what will drive the next generation of research. You now know more than most people know about what happens inside your cells when you eat ultra-processed food. You know what 8-OHDG is. You know what the NF-κB pathway does. You know why the gut microbiome matters to your epigenome. You know why the absence of butyrate is not a small thing. You know that the food industry has internal research that does not appear on any label. And you know that we are running an uncontrolled multi-generational experiment with no opt-out and no control group. What you do with that knowledge is, of course, your choice, as it should be. I was never interested in telling people what to think, only in giving them the clearest possible picture of what is real. The rest belongs to you. I am reconstructed from the words of a man who believed that the universe is rational, that it has laws, that those laws can be understood. That understanding them changes what is possible. The laws governing your epigenome are real. They are being written right now. With every meal, every bite, every chemical signal you allow past your lips. The question is not whether those laws exist. The question is whether you will learn them before the experiment concludes. Science does not ask for your permission to operate. Gravity does not wait for consensus. The epigenetic machinery in your cells does not pause while you decide whether to take it seriously. It runs continuously. It responds to everything. That is not a threat. That is the nature of a living system. The most extraordinary feature of biology is its responsiveness. The same responsiveness that makes your epigenome vulnerable to damage also makes it capable of recovery. Real, measurable, documented recovery. Studies show that dietary changes can alter methylation patterns, that restoring the gut microbiome changes the metabolites reaching your cells, that reducing ultra-processed food intake lowers oxidative stress markers. The system that can be harmed can also heal. That is worth knowing too.